Hello everyone and welcome to today's webinar, New Fixable Viability Dyes and Applications for Flow Cytometry. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit them at thermofisher.com. Now we encourage you to participate today by submitting any questions you may have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. Now, this webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain those credits. We will announce our two winners of the t-shirt contest toward the end of the webinar, so please stay tuned. I'd now like to welcome our speaker, Chris Langsdorf, a staff scientist at Thermo Fisher Scientific. Chris, welcome. You may now begin your presentation, sir.
Hi, thank you for joining us today. My name is Chris Langsdorf, and I'm a staff scientist in research and development at Thermo Fisher Scientific. And today we'll be discussing new fixable viability dyes and their applications to flow cytometry. Now, what is a fixable viability dye? Specifically, this is a fluorescent dye that allows us to distinguish live cells from dead cells. In this case, a fixable viability dye is able to enter through the compromised membrane of a dead cell and bind to proteins throughout the cell, while a live cell with an intact membrane excludes the dye, and so these cells have only dim fluorescence. And so as we see on the bottom right, we'd expect cells to have a bright positive peak and a dim negative peak, indicating the dead and live cells. And the distribution of these peaks is retained even after formaldehyde fixation, as opposed to those peaks on the left, as seen with propydium iodide, where the distinction between live and dead cells is lost after fixation. Now, most commonly, this type of dye is used in combination with multicolor immunophenotyping. And so typically, we try to choose a color of viability dye in a panel designer tool that will allow us to multiplex with the maximum number of antibodies to answer the questions of our experiment. And the reason we really need a fixable viability dye in a multiplex immunophenotyping experiment is that gating on live versus dead cells by scatter alone is not sufficient. There are actually dead cells within this live cell scatter gate. And we can demonstrate that by further gating on a fixable, live-dead fixable violet plot versus forward scatter, where we can see this theoretical live cell gate actually has live and dead cells within it. Now, those dead cells actually are known to non-specifically bind antibodies and can really dramatically skew the results of our experiment. As we see, if we look at only live cells, our distributions look as we'd expect. And so it is really critical, especially for high parameter experiments, to include a viability dye. And so this week, we're announcing the launch of 6 new fixable viability dyes. These range across the spectrum from the violet all the way out to the near infrared, and really fill underutilized channels on a flow cytometer. So in every case, they have the same performance you're used to, where live and dead cells are clearly distinguished even after fixation. And now with a total of 14 colors, you have even greater choice when designing a panel. And these panels can be bigger now that we have additional reagents, for example, in the very far red range of the spectrum. And so in the context of the existing fixable viability dyes, here's the list of all 14 with the 6 new reagents highlighted in bold. And so you can see really for any channel on a flow cytometer, there's probably a fixable viability dye that can be used or that can be added to a multicolor immunophenotyping experiment that you're conducting. And as we can see, regardless of the color, the distinction between live cells in the dim peak and dead cells in the bright peak is similar. We just have the opportunity now to install this viability indicator in whatever part of the spectrum best suits your experiment. And so as part of this launch of 6 new dyes, we have gone back and looked at the workflow, looked at the protocol required for these reagents. And so I'll show you briefly some of the expanded characterization we've done to really study these dyes. Now, first, the protocol that exists with the current live dead fixable dyes includes a low throughput protocol. And realizing that today many researchers are not just using a few tubes, but are actually doing medium throughput or very high throughput experiments, we've expanded the protocol included with these kits to include both of these methods. Now, these are similar in the fact that the first step is dissolving the dry dye in DMSO, and then essentially making a 1:1000 dilution onto cells. Then carrying out an incubation step, and then after that, I'll show you data to support this, but everything beyond this is we can consider optional. And so it is recommended to do a wash step after labeling your cells. Then it's possible to label with antibodies against surface antigens, then fix and perm if you prefer, and then label with antibodies against intracellular targets, followed by analysis. But again, everything in this box is now optional for these 6 new dyes. On the medium throughput method, essentially we do the same thing. We dissolve our dye in DMSO, and then depending on the number of tubes that you need to label, just dilute this dye 1:1000 into PBS, spin down your cells, and bring them back up in 1 milliliter of this ready-to-use working dye solution per tube. So this helps you scale a bit more quickly. For 96 or 384 well plates, essentially just spin down the plate and remove the media from your cells and add the working solution of dye to every well and proceed with the rest of the protocol. And so these reagents can all very easily be applied to higher throughput experiments. Now, I mentioned in the previous slide that among the things that are optional, one of these is washing. And this is somewhat surprising, but the data does support this conclusion that whether we wash our cells or we don't, whether we just label the cells and immediately analyze them without washing away unbound dye, we're still quite easily able to resolve live and dead cells. And in some cases, as in this NIR 876 example, there's really no difference between washed or unwashed cells. For absolute best results, it is still recommended to wash, but in cases where your cells may be sensitive to centrifugation, for example, it's not critical to wash your cells. Another interesting conclusion from our additional characterization experiments is that do you need to label your cells in PBS alone, or can you label cells with some protein present, such as bovine serum albumin? And we can say, once again, somewhat surprisingly, that we can label our cells in a buffer such as phosphate buffered saline with 1% BSA. And once again, in some cases, there may be a slight change in the signal or the background or both, but in most cases, there's very little change due to labeling in 1% BSA. And so in many cases, this will save a centrifugation step during your workflow. If your cells are already in a FACS buffer, such as PBS with BSA, you can just go ahead and label them and carry on with the protocol without a buffer change. And in the case of complete media, such as 10% BSA, I would recommend using either the NIR 780 or the Lyme 506. These both seem to be compatible with protein concentration up to 10%. With the other dyes, we will expect some loss of signal with that much protein present. But 1% is compatible with all 6 of these dyes. So another question that I often get from scientists is, if I label my cells now and fix and perm them, do I have to analyze them immediately, or is it okay to save them and analyze them tomorrow or on Monday after the weekend? And so in this experiment, I labeled cells with 4 different dyes, fixed and permed them with the eBioscience IC Fix and Perm kit, and analyzed them, and then put those samples in the refrigerator, in this case for 35 days, and analyzed the same samples again. And so as you can see, that distinction between live and dead cells is very well preserved because these dyes covalently bind to proteins within or on the surface of cells. And that is essentially largely unchanged over the course of a month. And so I'd say if you want to wait until tomorrow when your flow cytometer is available, or if it's Friday afternoon and you want to wait till Monday to analyze, the fixable viability dyes are fine with that. You might want to confirm with the rest of your antibody panel if it's compatible with that, but there's no concern about losing distinction between live and dead cells over time. Now, another potential source of concern is, are these dyes that I'm putting on my cells going to be toxic in themselves? That is, will these have some impact on the viability of my cells just by labeling them? And so to answer this, using the 6 new dyes that we're introducing, I labeled a population of cells with each of those and then put them back in an incubator for 24 hours and compared them to just DMSO alone. And then using another class of viability dyes called Cytox dyes, either Cytox red or Cytox blue, depending on the color requirements, we can see in every case our cells have the same background level of cell death, which is about 5%. So there's really no change between those cells labeled with DMSO alone for 24 hours, or with DMSO alone washed and then incubated for 24 hours, or those labeled with any of these dyes. And so we can say that these dyes are not going to induce more cell death in our cells of interest. Now, a final workflow example: when you're wanting to create single color controls for your multicolor immunophenotyping experiments, each of these dyes is compatible with our ARC amine reactive compensation beads. And with this bead product, essentially we put 1 drop of beads into a flow cytometry tube and label it with whichever color of dye we'd like a single color control for. After washing, we add a negative bead, and we have 2 peaks that can be used to very easily set compensation for these dyes without requiring the use of your cells. And so, as I mentioned at the beginning, these new dyes that we're introducing are designed to fill channels where live dead fixable dyes did not exist previously. And also they're screened so that they have minimal spillover into adjacent channels on a flow cytometer. And so, for example, using just the yellow laser on an Attune NXT flow cytometer, we can use live dead fixable orange in our YL1 channel to gate on live cells. And in this case, these are human PBMCs. So we'll gate on live cells, then gate on lymphocytes. And then we can use three additional antibodies, which are all PE tandems, to conduct a multicolor immunophenotyping experiment. And as we can see, there is compensation required, of course, but once applied, we can readily distinguish positive and negative populations of cells. And so in this case, with 4 dyes all on the same laser, we have now left the rest of our entire flow cytometer available for additional markers. So a few highlights we discussed, and actually one that we didn't discuss, is we'd recommend labeling with these dyes for 20 minutes to get the brightest signal. But in fact, in all cases, 2 minutes is often sufficient to get clear distinction between live and dead cells. So if your cells are very sensitive to being in the labeling environment, this is often sufficient, but we would recommend for the best signal to continue for the full 20 minutes. You can fix and perm, but it's not required. We also recommend washing after labeling, but again, that's not required either. So if you do want to sort your cells, this can be conducted as one of the last steps before that sorting. So let's move on now, and we'll discuss some applications, areas or experiments where the addition of these dyes brings some value to you. Now, often with a fixable viability dye, you may not see just a positive and a negative peak, especially if you're using blood cells, as in lysed whole blood or peripheral blood mononuclear cells. And in some cases, we may see 3 peaks. So to help decide which peak we should gate on, we can set a gate on the different populations and then back gate onto our scatter plot and see where these are. In this case, our dimmest peak is our lymphocytes. This intermediate peak is in fact our live monocytes, while these 2 brighter peaks are the live and dead lymphocytes. And so if we're interested in immunophenotyping all of our cells, we would probably expand this gate to include both of the populations on the left. If we're interested just in lymphocytes, maybe the gate on the left is sufficient. But in fact, a back gate will help answer which peak should you gate on. Now, another tool that will help answer this question is a new flow cytometer called the Attune CytPix cytometer. This is the same Attune NXT flow cytometer with the added benefit of bright field images of every cell that you have acquired. And so we can now set a gate on what we think is our live cells, look at a scatter plot and see that, in fact, these cells are where we would expect live cells to be by scatter. We now look at 3 representative images of cells within this gate. We see they're round and uniform and smooth, and as live cells should be. We now gate on the very bright positive peak and back gate to our scatter plot. These are where we'd expect dead cells to be. And we can see why the side scatter of this population is higher. They're quite granular. They have some irregularity and some texture inside. And so this is what our dead cells look like. Now, in this experiment, there was a 3rd peak in our viability histogram. If we gate here and back gate, we see these are dead cells, but also what might be called, beyond dead. These are starting to lose their structural integrity and fall apart. In that case, components of the cell that have been labeled with our viability dye are being lost. And so on a fluorescence basis, we'd expect these cells to be dimmer. And that's why this population is less bright than the rest of the dead cells. And so this is really why we see 3 peaks in this example. Now, the Attune CytPix instrument can also be used to show the interesting morphology of yeast cells as determined to be live or dead with live dead fixable orange viability stain. So if we gate on our negative peak, which is our live yeast cells, we see that these are in general a uniform circular shape, but in some cases we see buds emerging from the cells. And this indicates that these cells are live and actively proliferating, and via budding, they're creating new yeast. Those cells in the positive peak we know are dead, and with the bright field images provided by the CytPix instrument, we can see there's really irregular morphology here that creates higher side scatter, and in fact we can confirm not only that these are singlets, but that they are morphologically, they look dead. We can also look at other examples. For example, in immunotherapy, if we're studying chimeric antigen receptor T cells, very often these cells, after being produced, are cryopreserved for shipment or storage. And upon thawing, we'd expect to see a fair amount of dead cells within our entire cell population. And so by scatter, we can see this as two populations, which we could say are roughly live and dead by scatter. But as I demonstrated previously, a scatter gate really isn't conclusive. We need the distinction between live and dead cells as indicated by a viability dye. And so here we have some CAR T cells labeled with each of these 6 dyes and showing us a clear distinction between those live and dead cells. And so we can use that distinction between live and dead on an instrument such as the Invitrogen Bigfoot spectral cell sorter to show us a couple of things. One, we can visualize the entire spectral emission profile of this dye on a 7 laser Bigfoot cell sorter. But if we limit our view to just one fluorescence histogram of a single channel, in this case excited at 561 with emission at 575, we can now gate on live and dead cells and actually go a step further and sort out those live cells and remove the dead cells from our CAR T population. So in this example especially, it can be quite valuable to remove the dead cells from our entire population of CAR T cells and only carry on with our research or our analysis on live CAR T cells. So if we can look within our entire emission spectrum to one specific channel and display that a different way as a histogram, we can now sort out the cells in the negative peak and see that these are the live CAR T cells, while our dead cells, in this case we captured them, but we can just remove our dead cells from the mixed culture. If analysis is sufficient, each of these 6 new dyes is also compatible with other full spectrum cytometry instruments. So we can have a clear distinction between live and dead cells using whatever spectral analysis instrument is available to us. And while these dyes are intended for flow cytometry, of course, they provide very bright dead cells and very dim fluorescence in live cells. This distinction is also visible using fluorescence microscopy. And so, for example, using the EVOS M5000 imaging system, we can swap out the color of the LED light cubes within this instrument to match the spectral requirements of any of these dyes. And so we can see with 3 different examples that our dead cells are very brightly fluorescent throughout the cell, while our live cells have an absence of fluorescence. And so this distinction is actually visible with fluorescence microscopy as well. I will give the caveat that these are optimized for flow cytometry, and so with microscopy, A couple of these dyes may have photostability issues, and with the very long Stokes shift of some of the live dead fixable dyes, it can be challenging to find a microscope that is compatible with the unusual spectra of these dyes. But the LED light cubes contained within the EVOS imaging system can also be adapted to the Countess 3FL automated cell counter. And so with this instrument, we can take 10 microliters of our cell sample, put it into a slide, and ask this instrument to give us not only the concentration of our cells, but actually the percent dead in this case. So in this sample, we took 10 microliters of our CAR T cells, labeled with our live dead fixable lime 506 viability dye, and asked the Countess to give us the percent viable. In this case, we have 60% dead cells, and our cell concentration is 7 times 10 to the 5th per mil. And we actually have an image where it shows us which cells it's counting as live and which are being counted as dead. So I want to thank everyone for joining us today. I hope these new reagents will bring value to your flow cytometry experiments and hopefully give you some new ideas for what's possible, not only from a reagent perspective, but also with some of the really amazing new flow cytometry instruments that are available. So
thank you for your time.
Thank you for your time.
And thank you, Chris, for that informative presentation. We will now start the live Q&A portion of this webinar. And if you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many as we have time for. So Chris, our first questions coming already in from our great audience is, I would like to add a live dead fixable viability dye to my spectral flow panel. Can you provide any more guidance on how to design this panel?
Yeah, definitely. Thanks for the question. And this is a key concern now that we're seeing a lot of research pivot from conventional cytometry into the spectral space. And more specifically wondering how these dyes can be optimized to perform on a spectral cytometer. So first, I just want to mention that we have some great guidance available on our website on thermofisher.com. There's a full spectral cytometry page with guidance about designing panels for spectral cytometry. And also with our spectra viewer, the excitation and emission spectra of all of these dyes are available from 300 out to 900 nanometers, so you can really get a good idea of where some of these dyes show up in different channels. And also, just to mention, as we developed each of these live dead fixable viability dyes, we packaged them at a concentration where you get the best discrimination between live and dead cells. That is, they're at their brightest concentration. And so for conventional cytometry, bright is good. For spectral flow, brightest isn't always good. Sometimes having a unique and narrow spectrum is the best. And so to have good results on a spectral cytometer, for example, I showed on the Bigfoot or the Aurora, you may need to tighter these dyes down, that is, use a lower concentration, because we tend not to adjust the voltage on each channel on a spectral instrument. Instead, we'd prefer to use a lower concentration of these dyes to bring both the live and the dead peak onto scale without having to change the voltage. So that's one key piece of guidance is just consider once you decide on a color of dye to use, try doing a titration. Take it down in half or maybe one quarter of the dye to begin with, just to ensure that your dead cells are fully on scale and can be unmixed from your other fluorophores. So yeah, thank you for the question.
And thank you, Chris. What about fluorescent spillover in other channels in absence of a washing step for these new dyes?
Yes, thanks for that also. And I just will reiterate that it's surprising that we don't have to wash our cells after labeling with these dyes, and our standard protocol still does say to label your cells first with a live dead fixable viability dye and then wash, and then proceed to your antibody labeling and fixation steps. But when your cell model requires it, it's possible not to wash. And so in that case, the spillover to adjacent channels should remain largely unchanged. In the different histograms that I showed, in some cases we see a slightly higher signal in our live cells from not washing, but usually our dead cells are fully saturated to begin with dye, and so we'd expect their brightness to be largely unchanged. And also, the majority of users of these dyes are going to be gating on their live cells and then continuing on to gate on their various antibody markers in their panel. And live cells, we know, are dim and so have very minimal spillover into adjacent channels. So with these dyes, we're not so much concerned about the spillover of these dyes into to other channels. But I also see a good follow-up question to this that has just shown up, and actually how we can compensate these.
Sure. Did you want to go ahead and answer that question? The question is from our audience: how do we compensate those live dead dye?
Yeah. Thank you, Susie. And I think there are a couple of ways to compensate these live dead dyes, and compensation becomes critical as we get into these larger panels with many markers and many colors. So I showed one way to set compensation, and that's using our ARC amine reactive compensation beads that are designed to be labeled with amine reactive dyes such as these, and then just run as a single color control. And so we can use compensation beads for our antibodies, and we can use similar compensation beads for our viability dyes. However, as we begin to look at spectral cytometry, sometimes beads are not preferred, sometimes they are. If you prefer not to use beads to set compensation for any reason, it's also possible to take a sample of your cells and kill them. I generally just use 50 or 60 degrees Celsius for 10 to 20 minutes, depending on how sturdy your cells are, so that you can get a nice population of dead cells. Then we'll just label those with the viability dye, and that will give us a nice bright population to gate on. And this is often a preferred method for spectral cytometry, so that we can have our cells be consistent across all of our controls. So yeah, thanks for the question.
And thank you, Chris. And I want to thank our audience for these great questions. We have a couple more. Our next audience member says, could you put up the slide back up with the new dyes excitation details? And I know that we can't put our slides up right now, and I want to remind our audience that all of the slides will be on demand following our presentation. But Chris, did you want to talk a little bit about that slide? Maybe provide some information for that audience member?
Yeah, definitely. So in regards to the now that there are 14 different dyes, there are a lot of details as far as excitation and emission and where these fit into your instrument. And so we've built these details into a web page with all of not only the excitation and emission, but 3 different kit sizes. There's a sample kit, a medium, and a very large scale kit for each of these. And the easiest way to find these is just to go to thermofisher.com/livedead, and livedead is all one word, and that will take you to all of the information that I showed on these dyes, as well as a lot of other details on how to use them, a good protocol for their use. So thermofisher.com/livedead, or you can just Google live-dead fixable viability dyes, and it will take you there as well. So hopefully that clarifies, provides a good resource for you to find more information on those.
That's wonderful. Thank you so much. We have one more question. Are live dead fixable viability dyes compatible with the NovaFluor antibody conjugates?
Yeah, good question. And so as we begin to pursue much larger flow cytometry panels, the NovaFluor dyes, and especially NovaFluor antibody conjugates, are beginning to give us a way to really significantly increase our multiplexing ability. So these are new dyes that are intended to have a narrow emission spectrum and fit in quite nicely into larger panels. However, they are DNA-based scaffolds, and so other types of viability dyes, especially DNA-binding viability dyes, are not recommended for these. So things like DAPI or propydium iodide or cytox dyes are really not recommended for use in multiplexing with NovaFluor antibody conjugates. And so in contrast to those, all of the amine reactive live-dead fixable viability dyes work very nicely with NovaFluor antibody conjugates. And so I have a lot of experience in the lab using various colors of the live-dead fixable viability dyes in combination with panels using the NovaFluor antibody conjugates. And they make a good match together when you're pursuing larger, either conventional or especially spectral cytometry panels. So yeah, thanks for mentioning that.
Chris, I want to thank you for your presentation today and for your important research. Would you like to provide any closing remarks before we go?
Sure. Yeah, thank you, Susie. And I think just to reiterate, we had a great audience today. Thank you for all of the insightful questions. And of course, we value the feedback, and it's really the feedback in response to events like this that help us innovate the next round of new reagents and especially new instruments. So I encourage everyone to visit thermofisher.com/livedead and find more information about these reagents, and also the data that I demonstrated today on some of the new image-assisted cytometry and spectral sorting instruments is available on that webpage also. For further information, you can also reach out to your local Thermo Fisher Scientific representative for guidance on panel design using these reagents and really how best to use them and access them in your lab. So I want to thank everyone for joining us today, and we'll look forward to seeing you in the future.
And thank you again, Chris, for your presentation today. I'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And now I'd like to announce our two winners of the t-shirt contest, Miranda Batista and Fum Tak Akorakfane. Your t-shirts will be mailed to you. We would like to also invite you all to the next webinar on November 2nd, titled Fluorophore Fundamentals for Flow Cytometry. You can register for this event through the LabRoots website. Before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand with those slides, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Stay safe. Bye-bye.